There you go, folks. We're now live, okay? All right, so once again, any questions? Okay, let me share my screen with you, and we're going to jump right back into chapter 12. Okay, lots to do. Okay, so chat window is available, and some of your images on the video screens are also there so that I can see how you folks are doing. And the Microsoft Word document is up at this time. So I'm going to start with a question or two in the chat window just to see how well you are thinking about this content, and you know, if you're studying along, which I hope is the case. All right. Okay, folks, question in the chat window. What is the average resting membrane potential of a neuron? Okay, Elena is the first to contribute. Jamie, nicely done. Okay, thank you very much. Don't forget your units, right? The units do matter. So millivolts, okay? Minus 70 millivolts. Very nicely done. Okay. Let me ask you another question, something that we haven't explicitly talked about yet, but we will be talking about later this morning. What is the threshold potential that triggers an action potential? Okay, all right. So thank you, Alyssa. That's exactly correct. Again, don't forget the units, minus 55 millivolts, minus 55 millivolts. Okay. Final question. What is the maximum positive voltage that is reached during an average action potential? Okay, Alicia is the first to contribute, plus 30 millivolts. Okay, nicely done, ladies. Very nicely done. Folks, it is worth your time memorizing those particular numbers so that you have them in your mind as you think about this physiology, right? What's the resting membrane potential? What's the triggering potential, the threshold potential for an action potential? And what is the maximum voltage, positive voltage, that is reached during an action potential? These are critical numbers that you should keep in your mind. Okay, nicely done. And let's continue here with our conversation. We were talking about the resting membrane potential here. And so I'm going to continue off of this. Let's see here. Yes. So let's just start with this sort of statement, right? Simply documenting what we've already stated in the chat window. The average resting membrane potential for a typical neuron is minus 70 millivolts. Okay. There we have it, folks. So next, what we want to do is create a list of different factors that influence the membrane potential. Okay, what factors influence the membrane potential? There's several of them here, and if you want some visualization, I'm basically on page 418, 419 of your book at this time. Okay, so one of the major factors that influences membrane potential is going to be the unequal distribution of ions between the extracellular matrix and the cytoplasm of the neuron. Okay. The unequal distribution of ions between the extracellular fluid. I may have said extracellular matrix. That was a misspeak there. Extracellular fluid and the cytoplasm of a neuron. Okay. You know from reading your book that the extracellular fluid is quite abundant in sodium and chloride ions. The ECF, extracellular fluid, right? ECF, extracellular fluid, is abundant in sodium ions, which have a positive charge, and chloride ions, which have a negative charge. Okay. The cytoplasm has an abundance of ions as well, some positive, some negative. The cytoplasm is abundant in potassium ions, which have a positive charge, and in phosphate anions. And you know, because of that word anion, it's telling you it has a negative charge. The typical anion that we experience in the cytoplasm has the following formula: PO4 with a charge of -3. Okay, a charge of -3. It's kind of hard to write that online here. Let's make it a superscript. There we are. Okay. All right. Okay, so these anions, these PO4-3 anions, are typically attached to larger molecules like ATP and amino acids. Okay. The phosphate anions are typically bonded to other molecules like ATP and amino acids. And because of this, these negative charges are unable to actually leave the cell because they're bound to a molecule that can't simply diffuse through the phospholipid bilayer of the membrane. They're just too big. They're not going to get through. Okay. So let me note that a second factor influencing the membrane potential is the inability of most anions to leave the cell. Okay. The phosphate anions that are attached to other molecules are simply too large to pass through the phospholipid bilayer of the plasma membrane. Okay, they're just not going to get through. So they're constrained to be inside the cytoplasm. A third factor that influences the membrane potential is these sodium potassium ATPase pumps that move sodium and potassium ions against their natural gradients. Okay, so the fancy language that your book uses, okay, the fancy language that your book uses at the top of page 420 is this phrase electrogenic, electrogenic. Okay, so the third factor is the electrogenic genic nature of the sodium, potassium, ATPases. Okay, and what we mean by this, okay, electrogenic is simply a fancy way of saying that these ion pumps that are called sodium, potassium, ATPases have the ability to change the membrane potential by pumping the ions in the opposite direction from which they naturally diffuse. Okay. Electrogenic is a fancy way of stating that these special protein pumps, the ATPases that we're talking about here, have the ability to pump sodium and potassium ions in the opposite direction from their natural flow. Okay. And just so we understand what that really means is, let's be very clear about this. Okay. Okay, these sodium, potassium, ATPases, they actually pump 3 sodium ions out that have leaked into the cell, right? You remember from your reading and from the video I showed last time that there are these leak channels, right? There are these leak channels that naturally allow sodium to enter the cell and potassium to leave the cell. So if sodium is naturally entering the cell, you can expect the ATPases to pump them back out. Okay, so sodium, potassium, ATPases pump 3 sodium ions out, and in parentheses, that have leaked into the cell, while pumping 2 potassium ions in that have leaked out of the cell. Okay, so the sodium, potassium, ATPases bring sodium back out when it's inappropriately come in, and they push sodium in that's leaked out. Okay, so they go against the natural gradients. Okay. These ATPases move the ions in the opposite direction from the leak channels. All right. Okay, gang. Do you have any questions at this time? Okay. All right, let's continue. All right, so we're going to jump into some conversation about graded potentials and action potentials, and I'm going to show you a nice video on these topics. Okay. All right, let's start here. Graded potentials. Let me see here. Let's see. Yes. Okay. All right, so to get the ball rolling here, let me show you a video about these topics, because I think the video will help inform our conversation so that when we're documenting notes, you have this visualization you can rely on in your mind's eye to think about these things, okay? So remember from last time, graded potentials are used for short distance communication, like a few millimeters, and action potentials are for long distance communication, a few feet. Okay. All right, folks. So bear with me. I'm going to queue up this video for you. Okay. This is a WileyPlus video. And because this lecture is being recorded, you should be able to access these videos through the recording. Okay. So hold on one moment. Okay. Get things set up here for us. Okay. Here we go.
Membrane potentials, graded and action potentials. In order to describe the generation of changes in membrane potential, we will use a motor neuron, which stimulates skeletal muscle to contract. The neuron can be divided into three zones. The input zone, consisting of the cell body and extensions known as dendrites, has chemically gated channels in its membrane to receive stimuli from other neurons as graded potentials. These graded potentials move toward the trigger zone at the axon hillock. The trigger zone is where voltage-gated channels are first encountered in the neuron. If the graded potentials reach threshold in the trigger zone, then an action potential is generated. The action potential is transmitted via voltage-gated channels down the axon to the output zone, where it causes skeletal muscle to contract. In the input zone, graded potentials are generated when stimuli such as neurotransmitters attach to receptors of chemically gated ion channels, causing them to open briefly. Then, shift in the distribution of ions on either side of the membrane results in a slight change in electrical potential. Graded potentials travel a short distance through an input zone toward the trigger zone before fading away. It is the net voltage produced by graded potentials at the trigger zone that determines if the threshold of voltage-gated channels is reached to trigger an action potential. Some graded potentials occur when chemically gated sodium channels open to allow sodium ions to diffuse into the input zone. This electrically moves the trigger zone closer to or above the threshold. They are considered to be depolarizing graded potentials. If chemically gated potassium channels in the input zone are stimulated to open, it allows potassium ions to diffuse out of the cell and causes the inside of the membrane to become more negative, moving the voltage in the trigger zone away from the threshold. These inhibiting graded potentials are known as hyperpolarizing graded potentials. Since depolarizing graded potentials cannot individually reach threshold, an insufficient quantity does not cause a response at the trigger zone. If, on the other hand, there are enough of them to reach threshold, an action potential is generated. If there are more hyperpolarizing graded potentials than depolarizing, the net membrane potential in the trigger zone becomes even more negative than the resting potential. This would temporarily inhibit the generation of an action potential or require additional positive stimulation to reach threshold. When the net graded potentials arriving at the axon hillock reach threshold, the action potential begins. Both sodium and potassium voltage-gated channels are stimulated to open. Sodium channels open quickly, allowing sodium ions to diffuse in and add positive charges to the inside of the cell membrane. If enough positively charged sodium ions diffuse in to bring the voltage on the inside of the membrane to 0 millivolts, it becomes uncharged, and we refer to the cell membrane as being depolarized. Sodium channels are only open long enough to allow sufficient amounts of sodium ions to pass through to cause depolarization. At this point, the inactivation gate on the sodium channel closes to block any further inflow of sodium ions. In actuality, before the sodium channels close, enough sodium ions diffuse in to cause the inner surface of the cell membrane to become positively charged. The purpose of the inactivation gate is to prevent the membrane from stimulating itself and causing repeated action potentials. At the same time the sodium channels close, the slower voltage-gated potassium channels open and allow potassium ions to rapidly diffuse out of the cell. The removing of positive charges from the inside of the membrane causes its internal surface to become negatively charged again or repolarized. Once enough potassium has diffused out of the cell to cause the inside of the membrane to be below threshold, the inactivation gate of the sodium channel opens as the gates on both types of ion channels close, but not before the inside of the membrane becomes less than the resting potential of minus 70 millivolts to cause the inside to become hyperpolarized. Sodium-potassium pumps transport sodium ions out of the cell and potassium ions into the cell to restore ion concentrations and to, along with leak channels, re-establish its resting membrane potential. The depolarization of one area of the cell membrane causes neighboring voltage-gated sodium and potassium channels to reach their threshold. The result is the action potential spreads like a wave across the entire cell membrane. Once the action potential begins, all of the output zone will depolarize and repolarize. This is known as the all or none principle. In other words, the action potential does not diminish as it travels across the membrane, unlike the graded potential. So either all of the membrane depolarizes and repolarizes, or none of it will. It is also important to note that the amplitude of the action potential is always the same. It is the frequency of the action potentials that signifies a stronger response.
Okay, folks. So a lot of content there, and hopefully in the next 10, 15 minutes of note-taking, some of what you saw is going to become even more clear. That's my intention. So let me ask you at this time, before I start talking about these issues, do you have any questions about the video? Okay, this is heavy stuff. And I want to encourage you, in addition to taking notes, in addition to reading about it, it is super, super important that you truly answer questions that is in your textbook, right? So just as an example, don't forget that most figures have questions after them, right? At the bottom of figure 1214, there's a question. What kind of graded potential describes the change in membrane potential from -70 to -60, from -70 to -80, right? Figure 1215 on page 421. What parts of a neuron contain mechanically gated channels? The bottom right corner, page 421, there's checkpoints. What is a hyperpolarizing graded potential? What is a depolarizing graded potential? Those things are not just pretty things to gloss over and ignore. You've got to answer them if you're going to understand this content. So let me ask you a couple questions in the chat window, simply from having observed the video. Um, If the membrane potential of a neuron goes from +10 to -20 millivolts, what is the neuron experiencing? If the membrane potential of a neuron goes from +10 to -10, what is the neuron experiencing? Okay, so Elena's the first to contribute, and she suggests that it is repolarization. So let me ask you, do you agree with Elena? Well, to move us along, you should agree with Elena. Elena's correct. That is repolarization. So ask yourself, do you understand what repolarization is? Another question. If the membrane potential of a neuron goes from -50 to -100 millivolts, what is the neuron experiencing? Okay, Jamie's the first to contribute. Hyperpolarization it is. Nicely done. Nicely done. Okay. Final question. Will an action potential be triggered when the membrane potential changes from -80 to -60 millivolts? Okay, Jamie says no. So question in the chat window. Elena says no. Does everyone agree with Elena and Jamie? Well, you should, because they are correct. Okay, there will be no action potential as we go from -80 to -60, right? Because we need to reach a threshold membrane potential of -55. So tell me something in the chat window. What could further depolarize the neuron from -60 millivolts to threshold potential? What's going to have to happen to trigger threshold potential? Don't overthink it. You're doing excellent. You're doing excellent. Don't overthink it. What ultimately is responsible for the depolarization? Well, folks, to move us along. Okay, Elena says the inside of the cell has to be more positive. That's correct, Elena. Nicely done. And basically what that usually means is that there has to be an influx of sodium ions, right? Sodium ions from the extracellular fluid have to filter into the neuron, okay? So let me ask you one final question, right? These questions are designed to see if you're understanding, to see if you're really following along and not just blissfully taking notes and not thinking, okay? Okay, so you note, I'll make it a little bit easy for you. If you go back to page 417 in your textbook, page 417 in your textbook, okay? Let's do this. Question in the chat window. If you get smacked with a ball while playing ball tag, what kind of ion channels are opening? Okay, so page 417, page 417. If you get whacked with a ball while playing ball tag, what kind of ion channels are opening? Okay. All right. Awesome. Awesome. Everybody that's answered in the chat window has that one correct. Those are mechanically gated. Nicely done. Nicely done. Okay. And so just so we're sure, another question. What material will pass through an open mechanically gated channel to depolarize a neuron? Uh, okay. So, okay. All right. So I'm seeing a variety of answers here. And so the one that I'm really fishing for at this moment for the context of our conversation is sodium. Okay. Sodium is what's going to be doing it. Okay. Uh, Now let me ask you another question. When acetylcholine binds to the receptors on muscle cells at a neuromuscular junction, what kind of channel is opening? Page 417, what kind of channel is opening? Okay, all right, very good. Very nicely done, folks. That is in fact the ligand-gated channel, right? Uh, ligand-gated sodium ion channels where the ligand, okay, so let me ask you this. In our neuromuscular junction question just asked, what is the ligand? Okay, so we do see we might have a difference of opinion from some of the responses in the chat window. The ligand is acetylcholine. The ligand is acetylcholine. The ligand is a fancy generic word to refer to any substance which actually binds to a receptor. So acetylcholine is the substance binding to the receptor. So acetylcholine is the ligand. When it binds, the receptor changes shape. Sodium enters, depolarizes the muscle cell, and you generate a muscle action potential. Okay, I think that went pretty well from what I saw as responses in the chat window. And I hope for those of you that always stay quiet, I really wish more folks would participate, okay? Because I can't help you when I don't know what you don't understand. I hope you are understanding this. Okay, any questions before we take a boatload of notes? Okay, let's continue here. All right, so let's see. I'm not going to dwell on graded potentials very long, but I will put a couple things into your notes so that I know that you have it. Okay. Number one, you know that most graded potentials occur in the dendrites and cell body of a neuron. okay? You know that typically graded potentials will take place when a mechanically gated or ligand gated channel opens or closes. Okay. And so, you know, we have a few things that are taking place here, right? We recall that the resting membrane potential of a neuron is minus 70 millivolts, okay? Okay. So, if you have a stimulus which makes that minus 70 millivolt charge even more negative. So we go from minus 70 to minus 90. That's going to be called hyperpolarization. Hyperpolarization, okay? All right. If a stimulus makes the membrane potential, the resting membrane potential of minus 70 millivolts more positive. So we go from minus 70 to minus 20 to plus 10. Then the neuron is experiencing depolarization. Okay. 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 All right. Number 3. Okay. The term graded, okay, Graded, it means that the electrical signals vary in amplitude. Okay. Now, if you don't remember what amplitude is, take a look at figure 1216 on page 422, right? As you look at figure 1216 on page 422, you notice there that you see a green stimulus, a blue stimulus, and a purple stimulus. And you notice that the height of the bar is higher for the purple stimulus than it is for the green stimulus. That distance, that vertical height is amplitude. Okay, that's what amplitude means. The term graded means that the electrical signals vary in amplitude. Okay. Okay. Amplitude is the size of the vertical distance between the hump and axis of the graph of figure 1216. Okay. Figure 1216. Okay. The amplitude is controlled by the following. The number of channels that have opened or closed, and how long the channel remains open or closed. So there's a sense of time in this phenomenon. Okay. Now, final thing, and then we're going to move on to action potentials, right? Because these graded potentials only take place over very short distances, a few millimeters, they tend to die out very rapidly. But even though they die out rapidly in terms of distance, their strength and duration of time can actually be influenced by a phenomenon known as summation, which is when you have multiple graded potentials come together, they produce a larger amplitude effect. Okay, take a look at figure 1217, and it shows you the summation of the graded potentials. You see stimulus 1 there producing a small change, the first purple hump. Stimulus 2 comes in, produces its own change in depolarization. And you notice that when stimulus 2 overlaps stimulus 1, that the amplitude, the distance of the purple hump gets even larger. Okay, that is a phenomenon that is unique to graded potentials. It does not happen for action potentials. It only happens for graded potentials. Okay, because graded potentials act over a very short distance, they tend to die out quickly. But, okay, their impact can be influenced by summation, and I will refer you to figure 1217 to be sure that you appreciate this summation, okay? Let's see here. Okay, please note that graded potentials, that summation is a phenomenon that is unique to graded potentials. Summation does not occur for action potentials. Okay, action potentials work differently. Okay. All right, folks. So that's just some little notes about graded potentials. Okay. So again, somebody comes up, I know we're not supposed to be getting close to people, right? Social distancing and the whole 9 yards. But if life were normal and you're around people at Christmas time or Thanksgiving, you might see some old family friends. You go over and you give them a hug, right? You give them a hug because you're happy to see them. As they squeeze your body, mechanically gated sodium channels in the nerve fibers embedded in the dermal layer of your skin are going to open, and there's going to be graded potentials there that work on the dendrites and cell body region of those neurons. And if the summation is sufficient, that signal will eventually propagate to the trigger zone of the axon hillock, right? Coming off the cell body, between the cell body and the axon. That will allow threshold potential to be reached, minus 55 millivolts, and bang, we hit an action potential. Okay, so let's talk about action potentials. Okay. So, all right, so we are definitely on page 422, 423, and we'll be walking through this content here. As you know, most action potentials start in the axon hillock, and even more precisely, the trigger zone of the axon hillock, region of an axon, and spread along the axon. Okay. Number 2. Okay. Action potentials are what we refer to as all or none events. They're kind of like pulling a trigger on a pistol, right? If you pull the trigger on a pistol so that pistol ejects a cartridge, it fires a bullet, there's no way to undo that. There's no way to stop it. There's no way to bring it back. We can't reverse time and bring the bullet back into the barrel of the gun. That cannot happen, right? So when an action potential is fired, it's going to happen. It is an all or none event. Okay, action potentials are all or none events. Okay. Okay, we cannot. Okay, we can not unfire a gun. We cannot uncook an egg. Okay. All right, so let's continue here. Action potentials are going to be primarily made of two stages. Okay. Which you kind of know already. They are depolarization. Okay, depolarization is when the inner surface of the plasma membrane momentarily becomes more positive than it normally is. Okay, this is when the inner surface of the plasma membrane of a neuron momentarily becomes more positive than it normally is. Okay, and then this is typically due this is typically due to an influx of sodium ions. Although there are some scenarios where other positive ions can enter the cytoplasm. One of you mentioned calcium earlier in an answer to my question. That can do it as well. If you have any positively charged content that's entering the neuron, it's going to depolarize that neuron, okay? Typically, this happens when sodium ions enter the neuron. Okay, and so, you know, if we have sodium ions enter the neuron, you go from minus 70 millivolts to minus 55 millivolts. That's threshold potential. And then a whole sequence of other events will happen, which will rapidly depolarize the cell the whole way up to plus 30 millivolts. And we're going to walk through that. Okay, but here in our notes here, the second major portion of an action potential is the repolarization phase. Okay, this is when the inner surface of the plasma membrane becomes more negative again after having depolarized. Okay. Returns to a more negatively charged state. Okay. This typically occurs when potassium ions leave the neuron. Okay, so depolarization, sodium enters. Depolarization, sodium enters. Repolarization, potassium leaves. Okay, now there's other things that can be happening simultaneously, of course, but these are the major things that I want you to key off on, right? Depolarization, sodium enters. Repolarization, potassium leaves, that kind of thing. Okay. All right, let's continue. Number 4, action potentials are triggered when the resting membrane potential of minus 70 millivolts becomes less negative or more positive, right? Less negative and more positive is saying the same thing. Reaching minus 55 millivolts. Okay. So this is what figure 1218 is showing you there, right? Figure 1218 is really breaking down the various phases of the action potential. And so you see the stimulus there in figure 1218, the stimulus is there, and it's going to change the resting membrane potential from minus 70, and it's going to slightly curve up in the graph to minus 55. That's the threshold there, right? You see that along the right side of the graph. It says threshold, and bang, then we achieve a rapid depolarization phase. Okay, so examine figure 1218. Okay, number 5. Okay, never enough time here. Okay, unlike graded potentials, okay, the amplitude of action potentials is always the same. It does not depend on the stimulus intensity, not for an action potential. Unlike graded potentials, the amplitude of action potentials is always the same and does not depend on the stimulus intensity. Okay? So, if you were to very gently take your hand and very gently push into the cheek of your face, right? The amplitude of action potentials that allow you to perceive that you're pushing on your face are all going to be equal. However, okay, now if we went and we smacked the surface of our face a little bit harder, like that, the amplitude of those action potentials between gently pushing and smacking the face is still the same. Still the same. But what's going to be different between softly pressing your cheek and smacking your cheek is that the frequency of the action potential will be greater for the stimulus where you smack the cheek harder. So the amplitude is the same with action potentials, but the frequency changes depending on the intensity of the stimulus. Okay? So the question becomes, if amplitude remains constant, no matter how intense a stimulus is, how do we distinguish stimuli of different intensities? And the answer again is frequency. The frequency of the action potential changes. The more frequently the action potential occurs, the more intense the stimulus, okay? Frequency. The more intense a stimulus, the more frequently action potentials will occur, okay? So for example, okay, and this just is a very rough example here, okay? And I'm just playing with some numbers here to give you a very generic example here, okay? Okay, a gentle press on the cheek where you just gently feel that compression, you know, somebody's giving you a hug or, you know, something like that. You know, maybe, you know, just as a ballpark idea, maybe you get 10 action potentials in 1 millisecond. Okay. But if we smack you on the cheek, right, and it feels a sense of stinginess, wow, that hurt, that was more intense, right? If you get a smack on the cheek, maybe you get 1,000 action potentials in 1 millisecond. Okay, so the frequency is going to ramp up. You're going to get more action potentials in the same amount of time for an intense stimulus than you do for a weak stimulus. Okay. All right. Folks, any questions right now? Right? It makes sense now. Is it going to make sense on an exam? That's the challenge, right? Okay, so do your practice questions. Make sure you're doing the practice questions. Okay, let's step through a summary of the action potential. Basically, you can look at either page 423 or 424. There's a lot of text here, and I simply want to get this into your notes, okay? So number 6 here. A summary of action potentials. Okay, so we have step 1, which is the resting state. Step 1, which is the resting state, okay? So the resting membrane potential of minus 70 millivolts is largely maintained by the motion of sodium and potassium ions through the leak channels, Okay, the resting membrane potential of minus 70 millivolts is maintained by the sodium and potassium leak channels, right? The leak channels, right? It's like it's like floating in a rowboat and you have a leak in the bottom of the boat, right? It's a leak channel, okay? Okay. Please take note that at rest here, which is what we are talking about, right? At rest here, the voltage-gated channels are actually closed. Voltage-gated ion channels are closed at rest. Okay. And please take note that the voltage-gated calcium channels, which your book shares with you, actually have 2 gates, right? They have an activation gate and an inactivation gate. Take note that voltage-gated sodium channels have 2 gates, activation and inactivation. Okay, so that's step 1. We've got the resting membrane potential, which is generated and maintained by the leak channels, not the voltage-gated channels. Okay. So page 425 is probably a good visualization, figure 1220. Step 2. Okay, step 2 is the depolarization phase. Okay. When a stimulus, like we've been talking, the stimulus could be a smack on the face. It could be the odor of a skunk, depolarizing the olfactory nerves in your nose. It could be any number of things. Heck, let's be honest with you. You're looking at your computer screen right now. Photons of light are hitting the retinal neurons in the back of your eyeball, and they're depolarizing them. And that action potential is propagating through the optic nerve, across the optic chiasm in the brain, where you're learning about the anatomy, right? And you can interpret what you're seeing. Okay, so for depolarization, when a stimulus, okay, again, could be anything, an odor, a smack on the cheek, such as an odor, smack on the cheek happens. When a stimulus occurs, mechanically gated channels open, they open in such a way that in that moment of time, more sodium is entering the cell than potassium is leaving the cell. That's what accounts for the cells inside momentarily becoming more positive, which is what depolarization is, okay? Okay, in this moment, okay, more sodium ions enter than potassium leaves, okay? This changes the membrane potential from minus 70 millivolts to minus 55 millivolts, which is the threshold potential, okay? Folks, when threshold potential is reached, the activation gates of the voltage gated calcium channels open, and all of a sudden there is like that tiny little hole in the bottom of your boat. It opens up to a giant cavern, and all this sodium comes massively rushing in. This is a rapid depolarization phase, which occurs by the threshold potential changing the shape of the sodium voltage gated channel so that the activation gate opens and allows a river of sodium ions to enter the cytoplasm of the neuron, okay? At minus 55 millivolts, the activation gate of the voltage gated sodium channels. I guess I'll type the whole thing out here. Voltage gated sodium channels opens, and a flood, and the cytoplasm of the neuron is flooded with sodium ions. Okay, it's flooded with sodium ions. Okay, so folks, on page 425 of your book, do you see the difference between the activation and inactivation gates? Do you see the difference between the activation and inactivation gates? You see that there? Okay, all right. Now, obviously, this is all part of a large protein. Okay, okay. You know, I've got to ask you something that you can't answer, right? So here I'm going to ask you something. I'm going to challenge you. Can you answer my next question? Sorry, a lot of words here. Okay, there we go. The presence of activation and inactivation gates on the voltage-gated sodium channels implies that these channels are made of protein with what level of structure? Chapter 2, Chemistry chapter. Chapter 2, Chemistry chapter. Okay, all right, I'm tickling you, gang. Okay, but there really is a connection between things you learned at the start of the semester and things you're talking about now. Okay, the fact that you have these different gates is strongly suggestive of this protein having quaternary structure. Quaternary structure. Okay. So if you don't know what quaternary structure is, go look it up. Okay. All right. So let's continue here. I definitely got to finish this, and we're probably going to need another half of a lecture to get through neurotransmitters. Okay. Okay. All right. Let's continue. So let me just highlight here at step 2 that at the height of depolarization, at the maximum of depolarization, the membrane potential again is plus 30 millivolts. Okay, let's continue and get this finished. Step 3 is the repolarization. I'll say it as repolarization begins, just like your book says. Okay, so shortly after step 2, what we've just described in step 2, shortly after that, the inactivation gates of the voltage-gated sodium channels will close and the voltage-gated potassium channels will open, okay? The sum total of these physical acts is that the membrane potential starts to become more negative, okay? Shortly after step 2, the inactivation gates of the voltage-gated sodium channels close while the voltage-gated potassium channels open. This makes, in this moment, the membrane potential starts to become more negative. Okay. Step 4, and I'm pushing here so that we get this finished. Step 4 is the repolarization end. Okay. Eventually, the resting membrane potential is going to be restored, right? Because as repolarization continues, we're going to reach that minus 70 millivolt status again for the membrane potential. So at that point, the inactivation gates of the voltage-gated sodium channels open back up, and the activation gates remain closed, okay? And then we continue to maintain that resting potential through the activity of the leak channels, right? The leak channels are always open. They do not open or close. They stay open all the time. That's why they are leak channels, okay? Okay, so eventually the resting membrane potential of minus 70 millivolts is restored, and the inactivation gates of the voltage-gated sodium channels open up while the activation gates remain closed. Okay. Okay. Leak channel activity maintains the resting potential until the next stimulus is received. Okay. All right, folks. Let's see here. All right. I'm kind of in, I think we did pretty well here. So let me pause and ask you before I try to start, new material talking about synapses and neurotransmitters and things like that. Is there anything you'd like to ask right now? I mean, there's a world of content that I can ask you about on the exam. I mean, you see how involved this is. Okay. So is there anything that I can do to maybe help it be more clear to you? All right. Well, if something comes up during your study time, remember, I'm an email away and I still have office hours, right? Very few folks are taking advantage of office hours, and I guess it's because you have a lot of other busyness in your life, okay? All right, so let's continue. All right. Oh boy, okay. Rather than trying to cram in a video here, I'll cram in the video next time. And let me just do some notes about signal transmission at synapses, okay? Signal transmission at synapses. So I'm on page 428, 429. Okay, so we recall that a synapse is a region where communication occurs between a neuron and another cell, okay? Now we could generalize that, right? I mean, we are talking in chapter 12, which is the nervous system chapter. And so we talk about a synapse being a region of communication between a neuron and another cell. But honestly speaking, you could think of any two cells that are communicating as being a type of synapse. Okay, so when you have cardiac muscle cells in your heart that are joined together at the intercalated discs, and an electrical signal propagates through the gap junctions of the intercalated discs in your heart tissue. Well, that's a synapse too. That's also a synapse, right? So think about a synapse as a generic word which refers to a location where two cells come together to facilitate some form of communication. Okay. Number 2 and last note for today. Most synapses between axons are axodendritic. Okay, between an axon of one cell and a dendrite of another cell. But some are axosomatic and others are axoaxonic, right? So between axosomatic, between an axon and a cell body, axoaxonic, between an axon and an axon. All of these connections are possible and have been observed in nervous system tissue. Okay, folks, I'm going to stop right there for today. I'm also going to pause our recording because it's not necessary to record anymore.